You're in the Stem Cell Technologies Innovation Showcase. I'm Terry Thomas. I'm the Chief Scientific Officer at Stem Cell Technologies, and it's my pleasure today to introduce the speakers. The first speaker is Dr. Adam Hurst. He is a scientist at Stem Cell Technologies. He did his PhD in Peter Andrews' lab at the University of Sheffield. At that time, he focused on genomic instability in human pluripotent cells. He went on to do a postdoc at the University of Nottingham in the Albireo lab, focusing on primordial germ cell specification in non-rodent mammals. At Stem Cell Technologies, he's developed our new cloning supplement, Cloner, and he'll tell you more about that today.
Thank you very much for the introduction, Terry, and I'd like to welcome you all and thank you all for attending. So I'd like to jump straight into this kind of graphical outline of what we will be talking to you about today. So I'm going to show you how you can take pluripotent stem cells, how you can gene edit these cells, take this edited population, and generate clonal cell lines. And this is all encompassed within artesian media, workflows, and facilitated with our new product, Cloner. I'll show you how these clonal cell lines retain all characteristics of the uncloned parental line, and one of these characteristics is differentiation to all three germ layers. I'll then hand you over to my colleague and friend Enzo, who will take you through the new StemDiff cardiomyocyte system and how these cardiomyocytes generated using this system can be used for downstream applications. So I'd like to jump in straight away, and as many of you will know, working with stem cells, with human embryonic stem cells or induced pluripotent stem cells, they do exhibit low survival when you dissociate these cells to single cells and plate them down. Now this is a problem because of innovative techniques such as gene editing through CRISPR or other methods require the generation of clonal cell lines. And there have been a number of bottlenecks that have been identified during the single cell replating of PSCs. And these bottlenecks are survival post-plating. So when you seed the cells around 24 hours afterwards, you see extensive cell death. Of these cells that do survive, not many of them re-enter the cell cycle. And of the few that do re-enter the cell cycle, the survival post-division is very low. So you see many cases where the cell will divide and both daughter cells will die. So this all culminates in very low cloning efficiency, so typically less than 1%. And what's worth noting at this point is that if you have karyotypically abnormal cells or culture-adapted cells, these cells actually bypass these bottlenecks, ultimately culminating in a high cloning efficiency and increased plating efficiencies. So I'd like to introduce you to our new product, Cloner, and this is a supplement to enable the robust generation of pluripotent stem cell lines for innovative applications. Now, unlike competitor media currently available, there is no need for single cell adaptation with this supplement. And as a result of this, it's not selected for any karyotypically abnormal cells. So if we take karyotypically a normal cell line and clone this, the clones that we derive are all karyotypically normal. Cloner is also flexible. It's compatible with all artesian media, as well as multiple cloning matrices. You can see this, we're not stuck to specialist cloning matrices. We've actually tested some vitronectin, Matrigel, and laminin, and it works comparatively across those matrices. And the final thing is that it supports PSC clonal derivation using single-cell deposition, a technique which has until now been very inefficient, and you have to plate many plates to get a reasonable amount of clones to analyze downstream. So looking at some of the current methodologies out there, and I've chosen two examples here of what people are using. The first is low-density seeding, and this is the idea of plating multiple cells into one well. The advantage of this is you can generate a number of clones in one dish, so one experiment. However, the downfalls of this technique is there's no control over doublets, which you can see down and they will form mosaic colonies. And we all know that cells can migrate, find each other, find a partner, and form mosaic colonies. So ultimately, this technique it has its downfalls, and the success of this technique is largely dependent on the seeding density. If you seed the cells too high, you get an increased chance of these cells finding each other, forming mosaic colonies. If you seed the cells too low, the low cloning efficiency results in low colony yield, and you don't have enough clones to work with. So I'd like to kind of put up this example, and it's a question that I've asked a number of people, and I always get different answers as to how people are doing the cloning step. And these are two examples that I've heard more than once. And that's some people are seeding 15,000 cells into 10 centimeter dishes. I actually had one case where people are seeding 100 cells into a 96 well, and assuming that if one colony comes back from that, it's clonal. But I'm going to show you today how with Cloner we can seed just 400 cells into 10 centimeter dishes and still have enough clones to generate enough clones to use in downstream applications. So the second method I'd like to kind of discuss with you is single cell deposition. This kind of the gold standard of creating PSC lines. And it kind of negates these two disadvantages of low density seeding. So you can minimize the plating of doublets through stringent gating, and it actually places a physical barrier between cells so they're not able to migrate and form mosaic colonies. However, the impracticality of this is that the low cloning efficiency means that you get very low clones per plate. So if you assume kind of a 1 to 2% cloning efficiency, you're going to have to play a number of plates to get these done. And the problem with this is you're running the cells through fax machines. It's ultimately going to place extra stress on the cells. So if we do assume, kind of being generous, a 1 to 5% cloning efficiency in your PSC line, you're looking at between 20 and 100 plates to generate 100 colonies, which is kind of a good number if you're wanting to do gene editing or screen a number of clones. So I'm going to show you with Cloner how you can generate 100 colonies using just 4 to 7 96 well plates. So the workflow of Cloner is very simple. What we do is we take our PSC cultures, either modified or unmodified, and we harvest these cells to single cells. And the flexibility comes within using different matrices. So you can, whatever you have in the lab, you can use to do your cloning. We've used Vitronectin XF, Laminin, and Matrigel to do our cloning. And this is also compatible with MTESA1 and TESA-E8. So all you do with Cloner is you add the supplement between the first 4 days of cloning, whether you do this in 96 well plates or in 10 centimeter dishes. And you remove the cloner after 4 days and culture the cells for 7 to 14 days to generate your clones. And these are generally ready to be picked around that time. So this kind of illustrates the colony development throughout those days, and you can see by day 7 we're actually getting quite big colonies, and these colonies, if you wanted to pick them at day 7, you can. So these are the results from Cloner. So you can see on the left-hand side is I've actually got both of these two methods I've just described to you. So the first is this low-density seeding. And for this, I used around 25 cells per centimeter squared, which is around 10 times lower than the 15,000 example I gave earlier. And you can see we've done this here in TESA1 and TESA-E8. And you can see we've supplemented the media with either ROCK inhibitor, which gives us low cloning efficiency, and with Cloner here, which increases the cloning efficiency. Now this is across 4 different cell lines, 2 ES cell lines and 2 IPS cell lines. And you can see in both of these media, we do see an increased cloning efficiency. The graph on the right shows the single cell deposition, and this is the same, so just in MTESA1, either supplement with ROCK inhibitor or with Cloner. And with Cloner, we're hitting around 20% cloning efficiency by single cell deposition, so 1 cell per well. So you can generate around 20 colonies per plate. Now what's important to note with this is there's no single cell adaptation. These cells were just taken from a regular culture, a regular aggregate culture that we normally use, and all cell lines were karyotypically abnormal at the start of the experiment. And again, this was also seeded onto Vitronectin XF, so we're not relying on specialist cloning matrices. So with this, this experiment, I tried to sort of address what is a good cloning density, and people have different ideas of what this is, but I seeded these cells down at different densities from 800 cells per centimeter squared down to 20 cells per centimeter squared in 12 well plates on Vitronectin. And what was quite striking is that you can actually see that the cloning efficiency doesn't change across these different densities. So the more cells you see the same cloning efficiency. So if we look across this, we would need around 400 cells to generate 100 colonies. Now, if you take that 400 cells and you seed them into a 96-well plate, you can see that the density is very high. However, if you take the same 400 cells and you seed them into a much larger dish, so a 10 centimeter plate, you can see we're getting around 7 cells per centimeter squared, and we're still generating enough clones to work with downstream. And this slide kind of gives you a good visual of what I've just talked about. So this is a well-known technique and essentially published. So people seed 15,000 cells in a 10 centimeter dish. And you see here in ROCK inhibitor, we're getting very low cloning efficiencies, around 0.6%. You are getting some colonies back, but the chance of these being clonal are actually quite low. If you seed the same number of cells in cloner, you can see just how the increased cloning efficiency and how unlikely it is that any of these clones that you pick from this dish would be clonal. And if we kind of blow this up, you can see there's definitely colonies starting to merge within this. And in the ROCK inhibitor, you don't know, there's no way of telling really unless you have very good techniques of finding if this cell was a clonal, derived from a clonal single cell. So to illustrate this, what we did was we took the same cells and we seeded them down at 300 cells per 10 centimeter dish. And you see again here we're getting in ROCK inhibitor quite low cloning efficiencies, around 1%, with only 3 colonies coming back. However, if we seed the same cells into cloner, you can see around 27% cloning efficiency, and we're generating 82 clones from this. And this is at day 7, and the colonies are actually ready to be picked. So if I blow this up, you actually see that the colonies are quite robust, and you can actually pick these at this time. So I've told you kind of, it's all good to have a really high cloning efficiency, but what do these clones actually look like, and how do they behave once you've picked them? So what I did was I took the H1 cell line, and I used single cell deposition to derive clonal cell lines. I then expanded these and cultured them for 5 passages. So this is after 5 weeks in culture, and you can see the morphology is very similar to the parental line. So we have good ES-like morphology, and you can see here on the left-hand side is the right-hand side is the undifferentiated markers OCT4 and TRAL160 by FACS, and you can see all the clones express very similar levels to the parental line. One thing we were quite careful with when we were developing this product was we didn't want to be selecting for anything that was abnormal or grew faster. And as you can see here, the expansion rates following cloning in all these separate cell lines actually have very similar expansion rates over time. So we're not selecting for anything that grows quicker. And we also recloned these cell lines as well. And you can see we're not selecting for anything that clones from a single cell. So these are all, it's quite robust, and kind of to underpin this, all these cell lines that we generated are karyotypically normal following 5 passages. So all the cell lines are also pluripotent, so they differentiate into all 3 germ layers. So with this, we used our stem diff kits for ectoderm, mesoderm, and endoderm. And the graphs kind of illustrate the facts, so the clone lines I have kind of combined together, but you can see they have very robust differentiation to ectoderm, mesoderm, and endoderm, using the relevant markers, comparable to the parental line. And these just show ICC for these for relevant markers for this germ layers. So I guess now what I've shown you is how you can take pluripotent stem cells and derive clonal cell lines. But what I want to show you now is how you can do this step in between of doing the gene editing before you derive your clonal lines. So in this workflow that I'm going to kind of present to you, so this is kind of what we did. We designed our guide RNAs to a specific location. We used RNP, so we used protein Cas9 and RNA complex. We transfected this into our PSCs, and in this case we used an in-house iPS cell line, and we delivered this using electroporation. We then seeded these cells into plates to let the cells recover in M-teaser containing cloner, and then let these cells recover for around 48 to 72 hours, just to allow the gene editing to occur, which can occur between the first or second cell division. We then took these cells and we derived clonal colonies, and we also extracted genomic DNA to look at the T7 cleavage assay to determine the cutting efficiency. I then picked these clones and expanded them and characterized them for downstream applications. So in this example I'm going to show to you today, we disease modeled the long QT syndrome, or LQTS. It's important to note there are multiple genes associated with the LQTS, but we chose a gene called HERG, which is located on chromosome 7, and it represents around 25 to 30% of cases. Now, HERG2 encodes a pore-forming subunit of the potassium ion channel, and mutations within this gene result in abnormal ventricular repolarization. Now, there have been over 200 mutations identified in patients, and these happen within a heterogeneous autosomal dominant pattern of inheritance, so most patients are heterogeneous for this gene, and homozygous patients are largely lethal. So we picked chromosome exon 2 for the gene editing to try and knock this out using non-homologous end joining. So we formed RNP complexes, we electroporated them into the cells, and then we seeded these cells down just to recover in either cloner or ROCK inhibitor, and you can see in cloner we are getting increased recovery following gene editing, and you can see they're hitting around confluence around 72 hours. We harvested these cells and performed a T7 cleavage assay. And you can see in the fourth lane here, this is the cut band, and you can see we're getting around 30 to 40% cutting efficiency with this system. So we also took a portion of these cells and we seeded 400 cells into a 10-centimeter dish, and this is at day 14. And you can see with cloner, you can see we're getting around 32% cloning efficiency with 128 colonies derived using this system. What I did then was I picked 60 colonies into Emtesa or vitronectin, and 60 of these colonies made it to the first passage. So these are some examples here of the morphology we're getting at P1. So I then took a subset of these cells to look at the subset of these cell lines to look for the editing. And you see we performed a very simple cleavage assay to identify our heterozygote clones. And we picked one of these, we sequenced these, and we picked one of them to proceed with downstream applications. So this just shows the non-transfected control. Here, this is the wild type, and this is the cut site. And you can see it has the correct sequence. However, in the second clone, we found a 4 base pair deletion in one of these alleles, which results in a frame shift mutation, and Enzo will talk to you about the significance of this edit here. We also looked for non-specific binding, so the top 3 predicted target sites for this guide RNA, and all 3 of the top 3 all had good sequence alignment, and we didn't detect any off-target cutting. So what I've shown you so far is kind of this workflow, so taking your pluripotent stem cells, edit them, creating clonal cell lines, and how these cell lines are similar to the parental line, and how they can differentiate. And now what I'm going to do is hand you over to my friend and colleague Enzo, who's going to take you through the new StemDiff cardiomyocyte system and actually using these edited cells in downstream applications. Thank you.
The second speaker is Dr. Vincenzo Macri. He's a senior scientist at Stem Cell Technologies. He did his PhD at the University of British Columbia and went on to do a postdoc right here in Boston at Massachusetts General Hospital and Harvard Medical School. At Stem Cell, he's pioneered what we call the StemDiff cardiomyocyte system. It's first to market, and this system is media and protocols to cover the complete workflow of human PSC-derived cardiomyocyte. It includes the generation, processing, in other words, dissociation, replating, and freezing. He will tell you more about that today.
So what I'm going to talk about is our new StemDiff cardiomyocyte system and how we go from a PSC line or an edited line or a patient-derived line to make cardiomyocytes. And I'll talk about the applications and what you can do with those cardiomyocytes. So the StemDiff cardiomyocyte system is a media system with a number of components with 3 supplements that can take you through a differentiation sequence from pluripotent stem cells all the way through our 15-day protocol to generate beating confluent sheet of cardiomyocytes. And it's really made up of two components. We have the STEMDiff cardiomyocyte differentiation kit, which we just, for this talk, we'll call it the CMD kit. And then there's a second media kit, which is the maintenance media kit. And the significance of having the maintenance kit is that although I'll show you some data showing the quality control and the purity and yield at day 15, a lot of the application that's done in the field is taken on beyond that 15-day time point, where assays are run between day 20 and day 30 and even longer. So the maintenance kit really supports the health and viability of those cells moving forward, so you can use them for downstream applications, such as disease modeling, which we'll talk about today. I do also want to point that we did spend quite a bit of time figuring out how to process the stem cells before entering the workflow. This is a part of the workflow that is somewhat neglected in the field. There's not too much guidance on how you actually talk about generating these cells. So we've set up a way of a standardized seeding density to allow these cells to get to a confluency, and then at this point entering the differentiation protocol. So we go through several steps. We have supplement A that we use for 2 days, and then we switch to supplement B, and then we do 2 days of supplement C, another 2 days of supplement C to reach day 8. At this point, we have a highly confluent layer of progenitors, which then will transition into cardiomyocytes over the next 7 days at this time point. The cardiomyocyte system also includes a dissociation kit, so we generated a standardized way of dissociating, which is very simple. It's a 10-minute protocol. Again, the protocols for dissociation aren't standardized. There's many ways, but we focused on making it a simple and straightforward way, and I'll talk a bit about that. And then to finish off, the system also includes a support media, which is a transitional media that allows the cells to remain high viability during stress events, during harvesting, or coming out of a freeze-thaw. And we also have provided a freezing media so that the complete workflow, we have a number of media products to support the complete workflow. So why did we make the cardiomyocyte system? Why are we talking about it? So currently, there are some issues in the field. There's an efficiency of differentiation. So each IPS line that's generated, which is either edited or derived from a patient, or even isogenic lines from an ES line that you want to edit, there's variability in terms of the purity and the yield. So not every cell line behaves the same way in terms of your output of what the cardiomyocytes are. The quality and the viability of those cells, there is some variability with respect to harvesting and replating for downstream assays. So again, it seems like a trivial thing to do, to take these cardiomyocytes off the plate and seed them down, but viability is an issue, especially if you want to perform functional assays. So the solutions that I'm going to talk to you about today is the differentiation kit. So as I mentioned in the previous slide, we have a standardized seeding density that's applicable to a number of PSC lines, which I'll show you. We have a purity of greater than 80% cardiac troponin T, which is a specific marker for cardiomyocytes. Another number that we're quite happy about is that we get over 1 million cardiomyocytes. So from a single well of a 12-well dish, we're getting well over 1 million cardiomyocytes, which is a large number of cells that you can use for downstream applications. And for harvesting, replating, we developed a dissociation kit, which is a standardized protocol. It's a 10-minute method. And again, we're reaching numbers of greater than 80% viable cardiomyocytes. So even though you have 1 million cardiomyocytes, to process them and to use them, which I'll show you in a few slides, maintaining viability is important. So you want those cells to work and to be happy and to be functional. So how do the cells look like as we're transitioning through using our differentiation kit? So here is a quality HPSC culture that's maintained in our teaser products. This is maintained in teaser 1. And what we do is take these aggregates and we dissociate them using GCDR, our dissociation reagent for HPSCs, and we generate single cells. And we plate 350,000 cells down at day -2. and we let those cells then proliferate to confluency at day 0. And this is what the HPSC's monolayer looks like. So it's confluent, and this is how the cells should look like. When you're looking through the microscope down on your cell culture, it should be confluent like this at day 0. And this is where we add our first supplement A. And after 2 days, we move into mesoderm. So now the population of cells, which were PSCs, have now entered mesoderm at day 2. At day 2, we add supplement B, and at day 4, we now have a population of cells that have now entered cardiac mesoderm. And at this point, we add media C, and at day 6, we have an abundance of cardiac progenitors. Again, another application of media C to get us to day 8. And at day 8 is now when we see pockets of beating within that sheet. So here you can see it's confluent. The morphology has changed significantly as we're going through this developmental lineage pathway towards the cardiomyocytes. And at day 15 is where we do our purity and yield counts. You can see that there is a web-like and dense pockets of cardiomyocytes here that expand throughout the dish. So what does this look like in terms of a beating culture? This is an example of one IPS line. And at day 15, you can see here we have a complete, confluent, tightly packed sheet of cardiomyocytes. And this is what you will expect across a number of IPS or ES lines. So there's no gapping, it's confluent, and what's really neat about this culture is that it's synchronous. So we have a complete sheet that's beating together. So this is what you would expect. So in terms of its performance with respect to purity, again, as I mentioned, we use cardiac troponin T as our marker to designate the purity of the cardiomyocyte population. In an example here for an ES line and 2 IPS lines, we're getting well over 85% cardiomyocytes, and that's the troponin T is shown in gray here, and this is the isotype control in black. So we're reaching very good numbers at day 15 in terms of purity. With respect to the yield, and now with biological replicates of 3 examples here, again, the purity remains 80% and above across those 3 lines, the ES and the 2 IPS lines. But here, what we're seeing is well over 1 million cardiomyocytes. So we can get a range of between 1 million as kind of our minimum threshold of entry, but we can get well above that number. In some lines, we get 2 million. And with respect to the volume of media that we provide for the differentiation kit, one kit will generate well over 50 million cardiomyocytes. That's a large volume. So if you're interested in doing multiple IPS lines, if you're tracking a mutant through a family that requires a lot of IPS work, or if you're generating an isogenic lines like Adam had generated with, this is a lot of cardiomyocytes to work with, also for toxicology and drug discovery. So what they look like in terms of morphology, this is a confocal image. So we took that sheet of beating cardiomyocytes, we dissociated them with our dissociation kit, and we did a dilution, a very dilute replating of single cells. And so this is what you would expect to look like here. So this is about a 20-day-old cardiomyocyte, and here you can see that it's cylindrical, it's longitudinal, and it's stained with cardiac troponin T in green. And you can see the contractile elements, so they're striated, so it looks like a cardiomyocyte. Okay, so that's how we generate cardiomyocytes using our STEMDiff cardiomyocyte differentiation kit. So now let's talk about applications and how we can use these cardiomyocytes to push, science forward and discovery, and to figure out, what genes are doing. So now I'd like to talk about disease modeling, and as Adam nicely introduced, an arrhythmia called long QT syndrome. So what is long QT syndrome? So with respect to a surface ECG, if you look here at the bottom, a normal ECG waveform encompasses a P wave, which represents atrial excitability or contraction. There's a QRS, which represents ventricular repolarization and contraction of the ventricles. And then there's a T wave here, which represents ventricular repolarization. And what happens in long QT syndrome is that there's a prolongation from the time point of where the ventricles contract to when they relax. There's a lengthening of this QT interval, and we can see this here in the T wave, that the onset of that T wave is delayed, and then then as it's completing its waveform, it's delayed. And so if you look at a single cell of a ventricular cardiomyocyte, what does that look like at the single cell level? Well, this is a ventricular action potential, and here in black is what you would expect with a normal repolarization. However, with long QT syndrome, we see here that there's a delay in the repolarization, which maps with the surface ECG. So Adam mentioned that there's a number of molecular determinants which can lead to prolongation of the repolarization phase or the QT interval. However, in this example, we're focusing on the hERG channel. So we can see here, just represented here in blue, this is a hERG channel, it's a potassium channel, which pushes potassium outside of the cell, which allows repolarization of this action potential. When you have a disease mutation that's affecting the QT interval, we can see here in red that the total amount of outward current is lower, and that leads to this prolongation, why you see the delay and the lengthening of the repolarization. So why is this important? So this is one example. This is only one beat. But if you have an accumulation of these prolonged repolarization events, it can lead to instability or electrical instability of the heart, in particular the ventricles. And you can see here is a normal rhythm of 2 beats, but as the prolongation occurs, you can get these delays or these pauses, and then once the beat starts again, it can lead to a very bad instability. So this is called Torsades de Pointes, which is ventricular fibrillation. So there's an uncontrolled excitability, and this is the cause of sudden cardiac death. So long QT syndrome, which is not identified or not managed, it could have severe consequences. So we wanted to use the hERG channel as a model of repolarization and using it as a source to edit the cells using cloner, as Adam discussed earlier. So the wild type allele of the hERG channel sequence produces an ion channel. So it's a membrane-bound ion channel. And this is what it looks like here, composed of an N-terminus, the functional part of the voltage sensing domain, and then a pore-forming region. And this is where potassium ions move from inside to outside the cell, allowing the cell to repolarize. So what we did is targeted here the N-terminus, this pass domain. So the pass domain has been shown to be important in long QT syndrome. A family was identified about 15 years ago, and this family had a point mutation in this pass domain, and it was linked to long QT syndrome. So as Adam described, the 4-nucleotide deletion in this pass domain resulted in a truncation, and what that resulted is in an early stop codon in the N-terminus, essentially making that one allele null. The rest of the ion channel is not being made. So you have one working allele and one non-functional allele. So what we ended up doing was taking those wild-type control line and the isogenic mutant line and put it into our workflow. Again, preparing the cells for single-cell seeding and then entering the workflow using our differentiation and maintenance kits. And this is the example of what these cultures look like. Again, we're seeing a confluent layer of cardiomyocytes at day 15 for both the control and edited lines. Then what we did at day 18 is then harvest and replated those cultures using our dissociation kit. And then we replated them down on an MEA plate here. And what we ended up doing is then reseeded a high density of cardiomyocytes to restore the sheet of cardiomyocytes, not single cells, a high density, because with the microelectrode you need a lot of cardiomyocytes that pick up electrical activity. And so we allowed those cells to recover for 10 days, and then we measured excitability here at day 28. So just to revisit the cardiac action potential one more time. So this here illustrates the ventricular action potential or cardiomyocyte action potential. And there's a number of ionic currents which are responsible for this. So just to kind of, you don't have to memorize this, but just to kind of understand that there are a number of ionic currents which are controlling these things. And why we're talking about these parts of the waveform is just to explain a little bit about the output of the assay. So we're measuring a microelectrode array, an MEA. So it's an electrical signal, and it recapitulates the action potential that you would record from a single cell. So here is a spike amplitude representing here, so it lines up with the sodium current moving inwards. So that deflection is called the spike amplitude. And then you see this delay here, and that delay is in the MEA is called the field potential duration, the FPD, and that's composed of a calcium current and a number of potassium currents. And also another metric that we use is a beat period, so you can see the time between beats. And what we focused on here, again, is just now focusing on HERG. So there's a number of ionic currents that you could target, but we targeted HERG because it's involved in long QT. So what does this look like? How does the deletion, the early stop codon in the pass domain behave? So in black is the wild type control. So here you can see the spike amplitude. You can see a delay, the field potential duration. You can see the repolarization event here, this deflection. And then you can see another beat period of that cycle. So these are 2 cycles. So this is 2 beats, consecutive beats. In the HERG edited line, we can see that the deflection is, the spike amplitude is lower. But also what you see here is that there's a lengthening of this field potential duration. So the one allele is only working in the control, 2 alleles are working for that HERG. So here we can see we basically have recapitulated a delay in the repolarization event. And then also you can see that the subsequent beat is also delayed. So we've essentially have modeled targeting the pass domain and deleting that allele, the long QT phenotype in a single over across 2 beats. So what about consecutive beats? How does this look like? So now this is over a 15-second period. So we can see here that the wild type is beating quite stably. The beat is occurring earlier, and the phenotype of the HERG edited line is continuing. So we're seeing that there's a delay in the beat period and that the FPD is also delayed. So we're seeing less beats here. So what about long QT syndrome? So I showed you that the representative of the action potential is prolonged because there's a reduction in most likely the HERG current, and that is seen in a surface ECG. But what I told you is that consecutive beats is the problem. So if it goes undiagnosed and there are stress events or something that can trigger Torsades de Pointes or ventricular fibrillation, this is the problem. So can we actually see this in these cell lines? So now I'm going to show you about a 50-second recording from the control. Here we can see very stable excitability. All those metrics are there, they're stable. A spike amplitude is stable. The field potential duration is regular, and the beat period is regular. What about the pass domain, the HERG edited line? So we can see here now with the HERG edited line that the depolarization is delayed. And we can see here now that there is an instability in terms of the beat period and the frequency, and now we get this spontaneous rapid beating is what you would see in a human that occurs with long QT syndrome that's uncontrolled. So here we essentially have taken a control isogenic line for a controlled edited line and recapitulated long QT syndrome with respect to those parameters, the FPD, but also this spontaneous rapid beating. So we're quite happy. Well, it's unfortunate for people who have this, but we're quite happy to show that you can actually model long QT with our cardiomyocyte system and cloner. So what about disease modeling if you have patient lines? So it's great that you can edit and you can do all these innovative things, but there are also lines that you can have patient lines where you can track mutations in a family. So here we acquired lines from the Fraser lab at UCSD. And here I just want to talk a little bit about the phenotype and the genotype that's tracking through this family here. So this is 3 generations. And the little dot in the square here represents a carrier. So this is the founder mutation, and it's a point mutation in the hERG gene. And what you can see here is that the second generation of the 5 siblings, 4 have acquired this point mutation, and of the 4, 1 was symptomatic. So these are all heterozygous for this point mutation. And then we can see here the carrier 2-3, the mother, now has 2 daughters, and they both are carriers, but one is symptomatic for long QT. So I showed you the hERG channel, and we targeted the PAS domain. This mutation here is a single point mutation, a heterozygous mutation, and it occurs here at the C terminus. This work has been published recently by the Fraser Lab in Stem Cell Reports, I think just this month or last month. And we can see here that they did the relative expression of the alleles. We can see that all the carriers here have one allele that doesn't express the transcript. However, the wild types do. So what we ended up doing was the grandmother, who is not affected, so a control, and then the granddaughter, who has the point mutation but is symptomatic, and this girl's 18 years old. So we took these 2 lines, and using our differentiation medium, made cardiomyocytes. So we got the lines, we maintained them in teaser, brought them up, and then put them into our workflow to generate cardiomyocytes. So this is an example, replating those lines into an MEA, the same like we did with the isogenic edited lines. And here what we see, a confluent monolayer, and these are the electrodes, and this is the affected 2-1. So how does this look like? Can you see long QT in this family that's passing in a generational form, this point mutation? And we can see here in the black, again, just like the isogenic line, the black line is, the spike amplitude is large, the FPD is short, and it's regular. Here, with the affected granddaughter, who's the carrier of the point mutation and is symptomatic for long QT at 18 years old, we can see the spike amplitude is lower, and there is a prolongation at FPD. And strikingly, with this, in this case, for this point mutation, the beat period is much slower initially, but you can see here there's an instability. So it's very long, then it's short, and then it's long again. So these cells are electrically unstable. So I showed you 2 examples of modeling long QT with an isogenic edited line, or if you have a patient family that you're tracking a mutation, you can also use these cardiomyocytes for. Okay, so we talked about disease modeling with the cardiomyocytes. Now I just want to briefly talk about toxicology and drug discovery. How can you use these cardiomyocytes to do that? So I want to talk about using our medium for medium to high throughput toxicology or drug discovery. We're going to talk about toxicology. And what we did here is a little bit different. So this is a collaborative project that we did with Axion Biosystems, which was a very good collaboration, a fruitful collaboration, where we used their Cytoview 48 well plates here and their Maestro system, just like we did with the disease modeling. And what we did here was a little bit different. So we used the plates, and we did a direct differentiation using our teaser products. So we used teaser to generate our single cells, put them in the microelectrode array, and then enter the workflow directly into an MEA plate, and then record daily the excitability. So we recorded spike amplitude, FPD, and beat period from day 15 to about day 26 as a measure of excitability for these excitability parameters. And what you can see here is that we tracked in real time how those parameters change over time during differentiation. And at day 14 is where we can start picking up beat period. So with that confluent monolayer of beating cardiomyocytes at MEA, you really start seeing the beat period come on about day 14, and it starts to stabilize. And I want to make the point here that this is an experiment that we did with 4 different lines at the same time. So these are 4 PSC lines, 2 ES, 2 IPS, all done at the same time, all tracked at the same time, using the media together. And what's remarkable here is that the beat period and its stability reaches the same across all 4 lines. And then also the spike amplitude. So as I mentioned before, you want to recover a lot of, or generate a lot of cardiomyocytes so you can resolve signals here. So the spike amplitude with these lines are reaching 0.5 millivolt. So that's a size that's quite large, so you can resolve these metrics of FPD and spike amplitude and beat period. And again, here we start seeing it at day 14 and then tracking it over 26 days. We can see that it's, they're quite large. And again, the field potential duration, we can see here that at day 14, there's a little bit of variability, but then they kind of settle down at day 20 moving forward. So our cardiomyocytes, not only are they pure and high yield, and they look, in terms of the single cell level with a confocal, they're striated, and you can use them for disease modeling. They're functional, their excitability parameters are intact, and they're quite uniform across multiple cell lines. So what about toxicology? So if you're interested in drug discovery for any disease you're working on, every new drug has to be screened for hERG toxicity. So as I mentioned before, long QT syndrome is a problem if it's unchecked, or so you could have a long QT which is acquired or inherited, or you could have an acquired form. An acquired is through drugs. So if you have a drug that comes onto market, it needs to be screened for hERG, this one here. So if it affects this channel, you'll have prolongation of this, and you can enter into bouts of repolarization abnormalities and leading to long QT syndrome, and hopefully not the endpoint, which would be ventricular fibrillation. So here we used those 4 HPSC lines, and we used E4031, which is a specific blocker of the hERG channel. And here we can see that in black is the control. So this is before using E4031. And then after 10 minutes of application of 10 nanomolar E4031, we see a lengthening of this repolarization event. So the FPD is now prolonged. So that's what you would expect. So our cardiomyocytes are behaving and respond to cardioactive drugs, which you would expect. So, this is one example, but this is across all 4 lines. So we can see that on average, about a 40% lengthening of the FPD. So just to summarize what we just talked about, so Adam has described to you cloner, which can be used with our teaser product, and you can use that to derive clonal lines, and those clonal lines can be differentiated to all 3 germ layers. You can use cloner to efficiently generate clonal lines for gene editing experiments. Those gene edited lines can then be differentiated, and the example that we showed was cardiomyocytes. Again, I want to point that this is a specific example of cardiomyocytes that you can edit towards ectoderm or endoderm, so it's quite flexible. We're just highlighting cardiomyocytes today. And so with the STEMDiff cardiomyocyte system, we can now take those edited lines or non-edited lines and move through the developmental lineage stage of mesoderm to generate cardiomyocytes. And I showed you 2 applications of disease modeling and toxicology screening. So our media you can use for disease modeling and high throughput screening of drugs. So I want to just acknowledge there's a lot of people who contributed to this work. So this is our group in R&D that contributed to this work, but our R&D group is quite large. We have a quite active and collaborative group. We do great work in R&D. It's a great place to do it. Our product management team does an excellent job, managing our products and letting people know about them. And I want to mention Axion in particular, this group that would help with the MEA work and the analysis. So thank you.